see if you're satisfied with the thing. Point of information, I'm actually not a professor in Bern, but adjunct professor in Australia. I didn't get around to get habilitiert, you know that? Seemed not worth the trouble, since I already had that title. Anyway, psychedelic PET. I tried to find one of those droopy, melty psychedelic fonts that used to be popular, such as the cover of Jefferson Airplane Records, if that means anything to you. But I found this. It'll do. I'm not even sure why psychedelic letters and fonts drip like a melting Dali painting of watches, but I don't know. Anyway, psychedelic PET is the topic of my little presentation today, and PET, as I guess you must know, refers to positron emission tomography, which is a tool, an instrument, extensively used in nuclear medicine. It's really the core of nuclear medicine these days for diagnostics of diseases throughout the body, but its original application was for brain research until doctors found out how useful it could be, and then they kind of stole it from us. But that is what happened, and the principle underlying it I will get to in a moment, but I introduced this with the reference to the renaissance of research of all kinds into psychedelics action and its clinical applications in especially anxiety and depression disorders. This following about a 40-year moratorium that I believe is direct consequence of the Nixon-era war on drugs. And I think we have to concede that the war on drugs was lost because drugs seem to have won, in many countries at least, in the sense that the prohibitions seem to have failed and a more rational approach is finally prevailing, which means, among other things, that it's now once again a legitimate topic of research, whereas it had been rather marginalized for quite some time, only in a few isolated places where people were able to continue that research, notably, I think, Franz Wollenweider in Zurich. So in this lecture, I'm going to give a summary of what has been learned, mostly in the past 20 years, about the action of psychedelics, especially focusing on what has been learned through positron emission tomography, and skipping over other functional activation methods like fMRI-based methods, which have been very productive and fruitful in the hands of others, like people at Imperial College. But here I'm going to talk about PET, which is based on the principle of positron annihilation. The trick is to label a molecule, a biomolecule that is targeting something in the brain or in any other organ, according to some kind of classical pharmacological interaction, but carrying with it the kind of radioactivity that can be detected externally by positron emission tomography. And the principle is as such that the molecule contains incorporated into its structure this short-lived, on the scale of an hour or so, radionuclide that decays in an unusual mode of decay where it releases an antimatter electron called a positron into the medium of wherever the molecule carries it, which could be into the brain, for example. It releases this positron, which wanders some distance here, coming from the decaying nucleus, until it encounters an electron, and they meet in this, what do you call it, fatal attraction, I guess, of the positron-electron pair, which form a sort of an atom called positronium, which immediately collapses and goes bang, releasing all the energy that was bound up in the electron-positron pair, E equals mc squared, and that corresponds to an energetic photon called a gamma photon of about 512 kiloelectron volts, which is energetic. It will pass through lead, but it doesn't pass through detector crystals. And if you see the two yellow things, that represents the photons, the gamma rays, that are going off at just about 180 degrees from the site of decay. So wherever that decay happens, there's a vector described by the emission of the two photons derived from the decay. And if those two photons, or gamma rays, should collide with a pair of detectors, one on either side, that counts as an event, because it's possible to count that as somewhere along that trajectory there was a decay event. And here, just for curiosity, I think it's the first PET scanner from about 1965. It's much the same as contemporary scanners in its principle. It's a ring of detectors that are wired together so that synchronous events of two detectors going off in the same instant count as an event. And on the right-hand side, you see the principle, which is if that event takes place in the brain, it counts and registers. And by accumulating millions and millions of such decays, you can produce an image that shows the distribution of the molecule seen from the outside in a way that is essentially not at all harmful to the participant. So now we get to something you're familiar with. Now, my humorous story is that, well, you'll be the judge, but I had a mushroom identifier on my app on my phone, and I started putting my friends' photographs in it to see what kind of mushrooms they were. And for some reason, it identified me as this Kalkopf. I don't know why. I'm not making this up. But and psilocybin and the psilocybin genus of mushrooms and certain other species produce the psilocybin on the upper part of the chemical structure, which is not brain penetrating because it's got this highly charged phosphate on it. There's an enzyme in the liver that kicks that off, releasing the active metabolite psilocin, which then enters the brain. And you can see from the structure that it is an indole alkaloid, similar to, in some sense, to serotonin in its structure. And that gives an immediate hint as to what psilocin would be doing in the brain. Now, I alluded to the early work. Is 20 years ago early? I guess it depends. From your point of view, it is, yeah. Led by Vollenweider at PUC in Zurich. And he did a study that has yet to be repeated. I hope to repeat it with something else, with ayahuasca instead of psilocybin. And he used, in that study, the very popular PET tracer fluorodeoxyglucose, which, as the name suggests, is a derivative of glucose, radioactive because it's got a fluorine 18, this special kind of radionuclide, incorporated into it. And upon intravenous injection, it passes into the brain and is trapped metabolically in living cells in proportion to the metabolic activity of the nervous tissue. And so you eventually produce a picture by counting the events recorded from the PET scanner, shown in the lower figure as a representative image showing the accumulation of FDG, fluorodeoxyglucose, in the brain of a healthy control, in this case in a Alzheimer's disease patient who has a fallout of metabolism in the parietal lobe. Anyway, you see a pattern that maps to the distribution of metabolic activity, mainly neuronal activity. So you can imagine that if you scan someone at baseline and then give them a drug and scan them again, by comparing the two scans before and after, you can spot the difference and see where did the drug act in such a way as to have the net effect of altering glucose metabolism, in effect altering neuronal activity, which is the main consumer of glucose energy, and which is tracked by fluorodeoxyglucose. And so they did this study in, I forget, 10 or 15 subjects, scanning before and after administration of psilocybin at a psychoactive dose. I think it was 1 milligram per kilogram or so. And they see this result, that this figure shows the changes, the increases in metabolism that were observed after the drug. And in almost every brain region, the metabolic activity increased substantially, as if the psilocybin, or its metabolite psilocin, were driving the metabolism of the nervous system to an excessive or an increased extent, sort of between 10 and 20% increase. Now, many people have found this an extraordinary result, and it still, 25 years on, hasn't been replicated. Because it doesn't quite make sense. I mean, I can see no fault in the paper. It was very carefully done using very appropriate methods. But here's an example of a kind of why I think there's something going on that we don't understand yet. And this is another PET study where we see here the orange-colored or hot metal indicates regions where FDG metabolism was increased after a completely different drug, not a psychedelic, but a stimulant called fenfluramine, which is a very potent releaser of serotonin. So there's nothing that I know of that is more potently releasing serotonin into the living brain than this compound. And you can see it had some slight increases and some slight decreases in the brain of this group of volunteers, which doesn't quite jive with the Vollenweider observation of a global increase. So hopefully we'll resolve that before too long. It's an intriguing result, but it shows how much the field has lagged, you see. If I were to present on the effects of amphetamine as revealed by PET, there would be hundreds of articles, but there are only 2 or 3 articles on psychedelics so far. That will change. So could psilocybin really increase brain metabolism so much? Maybe so, but we have to dig deeper into this to figure out what's going on. So psilocybin, as you saw from the structure, or psilocin, resembles serotonin, and there's plenty of reason to think that its action in the brain, in the nervous system, is via binding to serotonin receptors, which belong to there are about 10 different kinds in the brain. But in particular, there's plenty of reason to link psychedelic effects to serotonin 2A receptors, which is a particular subclass of serotonin receptors. And that's the general belief, is that all psychedelics have in common an action at serotonin receptors. That's kind of a dogma. So serotonin 5-HT2A receptors are thought to be the common site of action of all psychedelic substances. Where are these receptors found in brain, you might ask? And it's pretty clear that from this image, which is the average of some PET scans that were conducted with a serotonin receptor ligand in Vienna by Rupert Lanzenberger group, and you can see it's a cortical binding site. We see in the sagittal plane that the whole cerebral cortex is lighting up with this stuff, especially maybe in the anterior cingulate cortex, which is responsible for executive function, and here in the visual cortex, which kind of fits with this visual hallucinations, perhaps. So that's where they're normally found, and nearly absent in subcortical structures. So probably frogs that don't have cortex don't hallucinate. We don't know. It's a good question. So again, Vollenweider, and again almost 20 years ago, did the appropriate study where he scanned volunteers before and after psilocybin, but instead of measuring glucose metabolism, in this study he used one of these serotonin tracers, similar to the one here, and subtracted before and after, from after. And the hotspots show where the psilocybin treatment reduced the binding of the PET tracer, mainly in the frontal anterior cingulate cortex and the frontal lobe and the orbital frontal cortex, which is here, where serotonin receptors are abundant. Interestingly, it didn't have much effect where they're also abundant in the posterior region. Now, what does this mean? Psilocybin has an affinity for serotonin 2A receptors. So the easiest interpretation here is that the psilocybin, non-radioactive psilocybin, is binding to some proportion of the serotonin 2A receptors, as expected, and they're making them unavailable to be detected by PET, because the PET tracer can't stick if psilocybin is sitting on it. Yeah? So this is called an activation or a challenge study, where the pharmacological challenge here with psilocybin reveals, in a before and after comparison, where the psilocybin is sticking in the brain, and it kind of fits with the serotonin 2A receptors. Now, this is a more recent result. The torch kind of has been passed with the retirement of Vollenweider. One of the main active groups in this kind of work is in Copenhagen in the lab led by Gitte Moos-Knudsen, who is producing a huge number of studies in recent years, studying specifically the actions of psychedelics. And they were also working with psilocybin and are measuring the plasma levels of psilocin, the active metabolite, in relation to that kind of occupancy study that I just talked about, where they measure the availability of the serotonin receptors as a function of time after giving the injection of the drug. Yeah? So any drug like psilocybin will have a temporal pattern to its effects. And we see that pattern in the left-hand figure here, where we see the actual individual plasma concentrations as a function of time extending out to about 6 hours after dosing, oral dosing with psilocybin. And you can see that there's quite a degree of variability, depending, I guess, on absorption. People There were also some high doses and low doses there. So there's kind of a pattern of this wave of psilocin in the blood, peaking at about 2 hours after oral administration and declining back to not much after about 6 hours. And you tell me that kind of coincides with the duration of the action of psilocybin on, it's the responses evoked by it fit pretty well with the blood curves, as they should. Oops. But on the right-hand side, they asked these participants, who were also getting a PET scan, as if it weren't enough, to rate the intensity of their experience, which they did. And you see some of them, the ones with the low plasma levels had less intensity of experience. It goes up to a maximum of 10, which is a way of saying you can't get infinitely high. I mean, you can only get so high. And it seems to plateau, you see. Probably at a certain point, any additional effect is, you don't notice the difference anymore. And you see that in some people, the peak effect, which is marked by ego dissolution and so on, lasted for up to several hours in some people with the highest dose. But that there was a lot of variability. More about that. There is something to this. It's not just experimental error. Some people respond more strongly to psychedelics than others. And that's an interesting question, why that would be. Now, I said that they also did PET scanning with the serotonin ligand, a different one than what we talked about before. Never mind the details, but you see on the left figure, what you should be familiar now, this very intense binding throughout the neocortex of the volunteers, and then scanned again while under the influence, and you can see that the serotonin receptors are much less available, not just in the frontal cortex as shown by Vollenweider, but globally, for reasons I can't explain yet, maybe just statistical artifact. And roughly speaking, red to blue means 50% less availability, thus 50% of the serotonin receptors in this group of individuals were occupied by psilocin, Psilocin being an agonist at serotonin 2A receptors is presumably exerting its effect via its interaction at the serotonin binding sites. And so long as the occupancy is taking place, the experience is experienced. They went further to plot in a kind of a classical, a variant of classical pharmacology where they correlated the individual plasma levels of the drug with the occupancy in brain, which was by design very low in the people who had a low dose and approaching 75 or 80% in the people with the highest dose. And it fits, the black line indicates a fitting of a one-site model, typical pharmacological calculation. So you can then calculate the EC50 and these kinds of things. And it's a very well-behaved ligand, as if there's a simple relation between the plasma levels and the brain occupancy. Now they went even further and correlated the occupancy with the intensity rating in this figure. And you see there's kind of a threshold. The intensity rating between 2 and 3 is hardly anything. It would be like a little bit of a buzz you might get from a coffee, I think. And that corresponds to an occupancy of about 40%. So having a 40% occupancy is hardly enough to perturb the system to such an extent that there's a strong experience. But you see these high scores of intensity rating suddenly appear when the occupancy exceeds 40 or 50%. That's interesting. So the psychedelic experience seems to be kind of a threshold thing. You have to get to some extent over and above, at least 50% over and above the normal activation of serotonin 2A receptors to produce the strong effect. And then in the right-hand study, they correlated the intensity rating with the plasma concentrations, and it also follows kind of a modelable curve with some more scatter to it, I guess, because, within each individual, they took several recordings. They measured the intensity and blood levels at 30 minutes, 1 hour, and so on. So there's more data points here. I think it's an excellent study because they really covered the whole gamut of plasma levels, occupancy, and experiential quantity, the intensity of the experience. And I think taught us a lot from that. The same group, also including David Eritzer, who's now at Imperial, went a little bit further in the direction of the psychological or experiential aspect of it. Rather than just rating the intensity of the experience, they took a specific element of the psychedelic experience, which is measured by some instrument of essentially the mystical experience, of merging with the universe or whatever. And it's not vague. There is a specific instrument, psychological instruments that they administer to get a number for the intensity of the mystical experience. And here, you've seen this before, it just shows the distribution of serotonin receptors at baseline. And in this figure, they show something interesting. Again, the intensity of the experience starts at 0, and it suddenly rises up to full intensity and lasts for about 2 hours, and then declines. And they took the slope, the duration of plateau of peak experience, and the rate of decline as kind of pharmacodynamic measures of how effective the psilocybin was, and did various statistical things. Interestingly, they compared the non-PET setting here in a more comfortable environment, here in the PET setting, which is a very uncomfortable environment, about as uncool as you can imagine, with clanking machines and white coats and very sterile environment, quite alienating. And yet it didn't cause people to have negative experiences in this study, at least. And furthermore, the kind of duration and amplitude of the effect was quite similar. So environment somehow does play a role, but for whatever reasons, the environmental effect on these measures was not so great. In clinical settings, they administer psychedelics in a comfortable lounge-like atmosphere, with music and things of that sort. And you would think that determines, in some measure, the experience. But according to these metrics, it didn't actually. Interestingly, they correlated in this study. They measured the baseline serotonin receptor availability, and asked, does your individual, because in this audience you're all, you're mostly about the same age, but even so, within the population, this population, there would be a kind of a Gaussian distribution of some people having more serotonin receptors and some people having less. So that would predict that could be a predisposing factor. Serotonin poor or serotonin rich people might respond differently to the same drug challenge. And that's kind of what they found. The people who, there was a negative correlation between the baseline level of serotonin receptors and the intensity, or peak measures of peak intensity and duration, plateau, and the time to return back to normal. Negative correlation with the mystical experience. So the serotonin status may determine how strongly an individual reacts to psilocybin, and account for much of the variability in that thing. That I find really interesting. And a little bit disturbingly, they have done all of these studies in young people your age, basically. And I've heard, I don't know if it's true, but I've heard that the psychotherapeutic effects of psilocybin, correct me if I'm wrong, may diminish with age, maybe because there's less serotonin receptors for the drug to act upon. That has to be sorted out. That's something that remains to be established. So, in summary, what they found is that your kind of baseline level of serotonin receptors sets the stage for how strongly you will react to the particular drug. In this study, some of the same people from the Gita Knudsen lab did not a PET study, but the equivalent of a PET study measured in pigs, ex vivo, where they gave the drug to pigs and then measured the receptors after some time had passed after a drug challenge, in control group and in experimental group that got the drug treatment. And I think I've made a mistake here. They measured two things in this study. They measured the serotonin receptors by so-called autoradiography, which is basically slicing the brain and incubating it with radioactive drugs and seeing how many receptors there are. And they also did another study using this funny compound, which is a measure of synaptic density. It binds to the vesicular transporter protein in neurons and in many neurons, and reveals in effect synaptic density. So you can see in this picture that synapses are most dense in the superficial layers of the cortex, in the hippocampus, and in a few other areas. And there's several belief in the field that part of the therapeutic effects of psilocybin may long outlast the acute effects. And there could be a substrate for that. They could be instructing the brain in such a way that it changes its nature, carrying with it a kind of a message that may persistently alter brain function. And insofar as psilocybin and other psychedelics may have benefits in anxiety and depression lasting months, it does sound like their plasticity is a good place to look. And what they found was that, indeed, this marker of synaptic density, called I can't even read it, UCBJ, increased in brain sections from the pigs by not a huge amount. And notice they only had 6 or 7 pigs, but the control pigs and the psilocybin pigs were different. And there was a kind of, especially in the hippocampus, a significant increase in synaptic density. Now that's interesting because synaptic density is a substrate, let's say, for plasticity. So the hippocampus has been taught a lesson by the single drug experience. I forget, sometime days or a week prior to the time when they were killed and their brains analyzed. They also measured the serotonin receptors using, again, an autoradiographic method. And and they found that, green is psilocybin. And this was 7 days after the treatment. You see that there was a decrease persistently, lasting 7 days, in the abundance of the serotonin receptors in the hippocampus and also in the prefrontal cortex. Oh, it was transient. It was pronounced at 1 day, and then it returned to normal. So there's a transient effect to decrease serotonin receptors, but going back to this slide, a persistent 7-day effect to increased synaptic density. So one model or way of interpreting this is that the psilocybin, when administered to these pigs, sent a signal by binding to the serotonin receptors in hippocampus and in cortex, and producing acute effects, which you also see after serotonin releasers like ecstasy, to decrease the serotonin receptor abundance by downregulation. But while that downregulation occurred, something else happened. By virtue of that increased signaling at the serotonin receptors, the message was sent to tell the neurons to increase their synaptic density, which might be a substrate for learning, actually. So that's also a very interesting story, and it begs to be repeated in humans, because they haven't yet gotten to that. In this case, they did the pig study first. Now we return back to yet another oldish Vollenweider paper where they stepped away from the serotonin system and thought about other targets whereby the psychoactive compound psilocybin could be influencing brain function. And here they did a PET study in human volunteers using something called carbon-11 raclopride, which binds to dopamine receptors. Now, they measured the binding in this typical before and after design to see how the acute administration of the psilocybin altered the availability of dopamine receptors measured by raclopride. Now, psilocybin doesn't bind to dopamine receptors at all, or scarcely at all. So it's not the same circumstance as in the previous paper by Vollenweider where they measured serotonin occupancy, or likewise the Danish studies. Here they challenged with psilocybin against a dopamine marker. And they found in this small group of people, but 6 out of 7 of their subjects had baseline dopamine receptor here, and then a significant 20 to 50% decrease. So psilocybin is somehow reducing the availability of raclopride or dopamine receptors in brain, and the usual interpretation for that, since psilocybin is not directly binding to the dopamine receptors but doing something else, maybe releasing dopamine by some presynaptic mechanism, and therefore the dopamine occludes or occupies a greater proportion of the dopamine receptors and creates the appearance that the dopamine receptors have disappeared because they're now engaged with dopamine. So I don't know what to think of this. It hasn't been much investigated. Everyone these days is focusing exclusively on the serotonin effects of psychedelics, But we have to, I think, cast a broader net and consider that serotonin may directly be acting, psilocybin is directly acting on the serotonin system, but it may by indirect means interact with dopamine system, and in terms of the magnitude of the effect here, it's similar to what you see with amphetamine, which is a massive releaser of dopamine, therefore occluding a large proportion of the dopamine receptors and producing this kind of decrease after amphetamine. Now, psilocybin isn't considered to be a psychostimulant, and doesn't produce the typical behavioral patterns that are evoked by amphetamine, such as increased motor activity. But something's going on, and in terms of a serotonin-dopamine interaction. Now, at that time, I was doing pig studies in Denmark with raclopride for dopamine receptors, and we thought it would be interesting to look at the effects of LSD on a very limited budget. Could it be that LSD, which is a notoriously dirty drug, psilocybin is fairly selective for serotonin 2A receptors, but LSD binds to practically everything, and it could be a good target for, exaggerate, it could be a good candidate for having, as part of the net effect, no doubt serotonin is involved, but it could be also binding to dopamine receptors. So I used that competition paradigm in the pig, and it's only 4 or 5 animals, so it's not very significant, but here's the dopamine receptors at baseline in the basal ganglia, very few elsewhere, and then at an hour after the LSD, I think it was I forget it was supposedly a psychoactive dose. They were anesthetized, of course, when they were in the scanner. So we don't really know how the anesthesia interacted with this. But you see, there's not really very much going on. And at 4 hours, which is still during the expected duration of action of LSD at that dose, there was sort of a decline. So not very compelling, I don't think. Certainly nobody in the literature has bothered to cite this paper, maybe for good reason. But it was a good try, I think, because we wanted to see what is an alternate target, and it's, I think, premature to ignore the effects of these compounds via the dopamine system. So this guy, that's the Sonoran toad, being hunted to extinction because of its secretion of its dermal glands, which is myo-DMT, 5-methoxy-DMT. And at that time, when I was doing pig research in Denmark, it occurred to us that we could label this. We could stick a radioactive carbon here on the side chain and make radioactive myo-DMT and test it as a PET tracer. And this is just a representation of the typical chemistry. You start with a precursor. This is called bufotein, which is also hallucinogenic, and incubating it in a reaction vessel with a radioactive substrate, you can then produce a new molecule that is myo-DMT, a very potent hallucinogen, actually dangerously potent according to some, I guess. Its propensity for producing overdose is well known. So that's how the chemists produce the molecule. It's always a matter of starting with a non-radioactive precursor and sticking a radioactive atom on it, and then injecting it in the pig. And here's where it stuck in the 3 pigs. And you can, if you remember anything about serotonin 2A receptors, it's a cortical receptor. Yeah. This is the cortex of the pig, and there's no binding. There's a hot spot in this corresponds to the nucleus accumbens, actually. Even though there's obviously no pronounced cortical serotonin receptor binding, we did go ahead and tried to block the binding with ketanserin in a classical pharmacological challenge to see if we could knock off the serotonin binding component with non-radioactive ketanserin, and it did nothing. So whatever the hotspot is, it's not serotonin receptors. It's the wrong place for them anyway, and I was never able to figure it out, what is going on with this drug. Myo-DMT failed as a PET tracer for serotonin system. Like many PET tracers, it failed. We never figured out what the hotspot is in the ventral striatum. It could be a subtype of dopamine receptors. But there's another issue, which is that tryptamines, like DMT and meo-DMT, do not have super high affinity. I mean, 100 nanomolar affinity compared to LSD, which has 1 nanomolar, they're approximately 100 times less potent. And that stacks the cards against using such a molecule as an effective tracer, because low affinity ligands mean that when you inject this tracer quantity, minuscule amount of radioactive meo-DMT, it doesn't even get a chance really to significantly bind. So it was probably, we don't know what's going on. I never figured that out. But around that time also I was interested in ayahuasca, and tried some analog of ayahuasca in the pigs, where I wanted to test the effect that a generalization of the so-called ayahuasca effect, which is the potentiation of one drug, DMT, via administration of a monoamine oxidase inhibitor, which blocks the first pass metabolism of the DMT. So you can eat DMT by the spoonful, and it won't do anything, because as soon as it enters the gut, it's immediately broken down by monoamine oxidase, the same enzyme that's also in brain cells. And but as you can see here, as you know, Amazonian Amerindians discovered that co-administration of an MAO inhibitor from one plant, Banisteriopsis, and mixing it together with another plant, the Psychotria, which contains the DMT, the MAO inhibition potentiated the action of the psychedelic substance, which would otherwise be ineffective. How on earth they hit upon that, I don't know. The shamans claim that the plants told them. And I'm prepared almost to believe that, because what are the chances of picking 2 species among the 50,000 plants in Amazonia? Anyway, here they are. You've seen them before, I guess, the vine, Banisteriopsis, and the which contains the MAO inhibitor, and the Psychotria viridis, which is one of the popular plant sources for DMT. There are others. Cooked together in a cauldron to produce the mixture that, when consumed, produces this intense experience, a visionary experience that in the hands of some has curative powers. Apparently, there's reason to think that it may be helpful in cessation of, in drug dependence, and also in having lasting effects on anxiety disorders and depression. Remains to be fully established, but they're going full speed ahead along this lines in Zurich at Puck to develop a formulation that is a chemical equivalent of ayahuasca, containing the pure substances DMT and harmine, which may or may not actually reflect the true nature of the traditional mixture. And they're proceeding to trials, having first established the safety of this preparation, containing, on the left-hand side, the MAO inhibitor harmine as a pure substance, and on the right-hand side, DMT, which they're actually isolating from mimosa as the source for it, but it's the same molecule, combined together in a tablet for sublingual administration. We're going to test that, hopefully, in the fluorodeoxyglucose PET paradigm in volunteers when we finally get all the permissions to do this study. But just to remind ourselves of what's going on, here's the harmine, which is the MAO inhibitor that can be metabolized by hepatic enzymes of various kinds. But while it's in the blood or in the body, it blocks monoamine oxidase very potently, monoamine oxidase A, which therefore inhibits the breakdown of DMT to this acidic compound called 3-indole-acetic acid, which is inert. And so long as there's MAO inhibition on board, that main pathway for metabolism of DMT is blocked, and the other pathways could be continuing along, but they're not nearly as high capacity as the MAO pathway. To the first approximation, DMT metabolism is via MAOA. So we wanted to get at this. This was a work with my PhD student in Zurich, Clemens, who might be known to some of you. And we went to Denmark to do kind of a pilot study, which we're finally, hopefully, will see the light of day before too long, where groups of rats were given either nothing, saline, or harmine, or DMT, or harmine plus DMT. So it's a 2x2 design, yeah? And Then they were injected with various radioactive drugs. Some of them were PET scanned against FDG, and we did various other analyses of the brain tissue to kind of correlate to confirm the putative action whereby harmine would be potentiating the uptake of subcutaneous DMT. And this shows the FDG PET results, where we have in each column at different levels the control animals, the harmine alone, the DMT alone, and the harmine plus DMT. There's no precedent really, but for the paper by Vollenweider with psilocybin in humans, and there they saw a substantial increase in glucose metabolism. Here we see almost nothing, except maybe one spot. So it doesn't look like this particular drug regime had really any effect on glucose metabolism in these rats. But you could say it's kind of a pilot study because there were only 6 animals. However, I think we could have seen if there were a substantial increase, even with that small group size. So in that pilot study, we didn't see much sign of altered glucose metabolism from that dose of harmine DMT, 1 milligram per kilogram each, which should be a psychoactive dose. We didn't assess that behaviorally in the rats. But for this, we still don't have an explanation why the discord with the human result. It could be a species difference. Time will tell. We also measured in the brains, post-mortem of these animals, various metabolites. And the key result here is that in the 4 groups, control, harmine, DMT alone, harmine plus DMT, only in the harmine plus DMT was there significant brain concentration of DMT. If you didn't give MAO inhibition, there were masses of the DMT metabolite formed in brain, which was reduced by 50% by co-administration of harmine. So in other words, we've sort of confirmed what was probably fairly obvious, that DMT just simply doesn't get into the brain, or it does get into the brain, but it is immediately broken down to its inert metabolite unless you block the monoamine oxidase enzyme by co-administering the MAO inhibitor in the form of harmine. And we see several other metabolites and things going on there that I could talk about. So that kind of confirms the effect. 1 hour after the DMT administration, only traces were present in the rat brain, but it was as if as soon as it got into the brain, it was immediately broken down by MAO to form this metabolite. But when given together with harmine, the MAO inhibitor, we got sort of micromole concentrations of DMT in the rat brain, measured an hour after the treatment. Micromole is, I would call, a high concentration. Even if DMT doesn't have very high affinity, it should be sufficient to have interacted with the serotonin receptors in the brain. Did it though? Well, we didn't do the PET study. We did an ex vivo study where we gave a radioactive serotonin ligand, and then after various treatments such as harmine DMT and harmine plus DMT, and measured the availability of the sites. And if there had been a significant occupancy by DMT, for example, in this group where we know that there was high concentration of DMT in the brain, the binding should have been reduced. So we weren't able to prove or confirm an interaction between DMT and serotonin receptors. Reviewers are not liking this, let's put it that way, because it should be binding to serotonin receptors. And it is, I'm sure it is, but there's something we're missing with the time course, species difference, or something which we haven't nailed down yet. Because the other thing that's lacking or deficient in this study is that we didn't assess behaviorally whether the drugs were having an effect. Like we could have measured certain behavioral indices of the animals to ascertain that this combination of DMT plus harmine actually altered behavior, but whether or not it was a reasonable dose and it got high concentrations of DMT in the brain, and yet, even with that high concentration, it didn't block the serotonin receptors. No discernible occupancy. So I'm almost at the last slide here, and I'm sure you're almost at the end of your patience. So stepping back to something really classic in the PET world, which is work from the pioneering PET research group of Brookhaven National Lab in upstate New York. And they were doing early on measuring MAO in the brain monoamine oxidase, and in the different organs of the body. And you can see that there's quite a lot of MAO in the brain. There's some in the myocardium, a lot in the kidneys, and if you could see the gut, it would also light up here. So that's where the MAO is. They, in doing their study, scanned a smoker, because 30 years ago people in Brookhaven still smoked. And they found this surprising result, and only afterwards realized it was because, likely because of the smoking, this person had practically no MAO activity to be measured. Something in tobacco smoke is blocking MAO in the brain. It's probably harming itself, because Banisteriopsis obsessivine is not the only plant that contains harmine. A lot of plants do. Furthermore, it's formed in tobacco smoke by pyrolysis of tryptophan. So probably oral tobacco doesn't have MAO inhibition, but smoked tobacco and vaping would probably not have MAO inhibition. But smoked tobacco exerts this very pronounced effect of blocking MAO. And I've always been curious about that. The Brookhaven group showed a global reduction of binding to actually both kinds of MAO in smokers. And the MAO-A part was certainly due to, is likely due to the harmine that's produced in combustion of tobacco. And so it's quite a significant effect. You can see it's clear that there's substantial blockade. What would that do? Well, what does MAO do in the brain? It's responsible for the breakdown of serotonin via MAO-A and dopamine via MAO-A and B, the two variants. And so with that degree of serotonin of MAO inhibition, it's reasonable to expect that the brain concentrations of dopamine and serotonin have increased. And of course, we can't measure that directly in people, but you can do that in rats. So blocking MAO should increase the amounts of dopamine and serotonin, therefore potentiating or amplifying the effects of nicotine when co-administered in tobacco smoke, you see? It's plausible that everyone has said that tobacco is purely a delivery vehicle for nicotine. If that were the case, then patches and gum would make smoking unnecessary. It didn't work. There's something else, also conditioned aspects of smoking. But I think there's an argument to be made that MAO blockade is contributing to the psychopharmacology of tobacco. And could smoking be ayahuasca light? Yes, I think it is. Except people haven't really looked at that because smoking is not actually hallucinogenic. But I'm pretty sure that the MAO inhibition experienced by smokers is altering their responsiveness to nicotine. And I believe that's all I had to say. That's, how did I do for time? Hey, that was not bad. 45 minutes, yeah? Okay. I think I've earned my apero, yeah? But before we get to that, if you have any questions, please feel free to fire away. No? Please, you first.
I was wondering in the study where they compared like good and scary environments, so to say, if maybe the sterile environment wasn't that uncomfortable for the participants because they knew they were in a clinical study and felt safe.
Yeah, and these are people who are highly motivated, curious about learning about how these things work, and also there are people with some previous experience with psychedelics. So when I get to finally get the final permissions and start scanning with FDG, we're only going to recruit people, volunteers from Zurich, who have already been through the mill and are probably able to tolerate the experience. And yeah. So, I mean, environment is surely doing something, and everyone seems to believe that a positive environment, and as was discussed by several speakers at a recent conference in Bern, something about nature seems particularly, it's we don't know why, but something perhaps about the depth and the degree of interconnectedness that is inherent to nature may promote positive experiences. But as you see, there's not a lot of nature in nuclear medicine department. You could put a potted plant in the corner, I guess. Yes.
I think the thing about tobacco is really interesting. You said that a year ago also on in the Alps for
Yes.
and I really wondered the connection because you have in Amazonia the reverence for tobacco is so great. It's called like
Yes.
the master plant. So it's easy to think that this is something to do with on a pharmacological level with the blocking of the mouth. But then again, if you say now it's only working via smoke, but then different ethnic groups use it in liquid form or as a snuff.
Curing may be a factor. I don't think it's really known yet because I haven't seen the report where they measured the harmine content or other beta-carboline, harman and harmol, in different preparations of tobacco. One informant a researcher tells me that in tobacco smoke it's mostly formed from pyrolysis of tryptophan. But for all we know, in various curing procedures may also generate the harmine or other molecules like that. But yeah, it's true. You know, Ilana, my postdoc, Ilana Berlowitz, is more on the tobacco side. Her work is with tabaqueros in Amazonia. And she points out that it may be a simplification to view ayahuasca as this binary mixture of MAO inhibitor and DMT, because depending on where one is in Amazonia, and depending on local practice, she tells me that sometimes they don't contain any DMT. Mm-hmm. In fact, sometimes it's pure Banisteriopsis.
Yeah.
And also, furthermore, they add tobacco to it, plus some belladonna alkaloid. You know, just it could be. I guess it's a bit pejorative to say witch's brew, but it can be variable composition. It's not a standard pharmaceutical product. So there is additional layers of complexity. Yeah.
But do you know if there's any interest in pursuing the research into combining DMT with this any kind of like the MAO inhibiting?
Oh yes, in Zurich,
function?
that's their thing. They have developed a formulation specifically containing harmine and DMT in the correct balance to get a sort of an optimal DMT effect.
Yeah, but in combining it with the smoke or the tobacco.
Well, this is where it gets interesting. If their participants who are heavy smokers should have be preloaded with MAO inhibitor.
Yeah.
And as far as I know, no one has looked to see, if tobacco consumption, habitual tobacco consumption, alters the responsiveness to DMT or other substances. It seems to me that it should be a factor. I mean, it's hard to discern because apparently tobacco use is no longer common or is not common among ayahuasca participants. So you'd have to do a study where you had smoking ayahuasca participants and non-smoking, or something like that.
Yeah.
On the other hand, oops. Oh, that was rude. I didn't mean to do that, sorry. I'm trying to find this paper, where they found a threshold for serotonin occupancy. I mean, it's just an analogy, but with less than 40% occupancy, there was hardly any effect. Similarly, or analogously, a 40% MAO blockade may not be enough to potentiate the action of other drugs. When given therapeutically as an antidepressant or an anti-anxiety medication, MAO inhibitors, they generally aim for complete blockade. So there may be some threshold phenomenon that you really have to hit MAO hard to overwhelm because it's essentially superabundant. There's an excess of it. Yeah. Yes.
Maybe quickly, what I know of, they're doing a new study in Zurich, the group in Zurich with DMT and harmine, and I saw the screening criteria, and they ban habitual smokers.
Yeah.
Okay.
So it's maybe a side effect.
Yeah, I'm not sure if they banned it because of this argument, but just for general wanting to simplify and reduce the number of variables. Yeah. Yes.
There was one slide that showed the synaptical density that increases in pig brains.
A marker for synaptic density, a particular protein that is expressed in synaptic vesicles. Might not be exactly the same as synaptic density, which would be an EM thing, how many synapses there are per cubic millimeter. Yeah.
The follow-up there was only 7 days. Are there any studies with a longer follow-up?
Not yet, no. But you can bet that people are working on that. The claim or the belief is that the effects can be very long-lasting, that the serotonin effect is only short-lived, but it leaves an imprint in some sense on the brain. Yeah. But it'll take more research to establish that. It was nicely going from left to right. No? Nobody in this wing? Yes?
Maybe a more basic question. What do you have to do to get the permission to do these kind of studies here in Switzerland?
Well, I think it was probably easier in the cowboy days 25 years ago when France was doing these studies. But he, it is regulated. You can't just cavalierly inject radioactive drugs and intoxicating substances into people. Here, we had to, actually, Alana Berlowitz spent a long time writing and rewriting the proposal to the Ethics Commission, which is organized on a cantonal basis. And, they sent it back several times for adjustments. It helped. I think it helped that the group in Zurich have already developed this compound and used it safely in a large number of people. So precedent helps. I've now obtained the ethics approvals a few months ago. The next stage is because it involves administration not just of a radioactive tracer, the FDG, which is not very problematic. I mean, they do 10 scans a day with that here in the clinic. But we had to get approval from the BAG, the Bundesamt für Arzneimittel, and that's still in progress. It'll take a couple more months. So it's in stages, but the first thing is you have to get ethics approval, and then you, it's tricky, and it took longer than I expected, and that might be partially because I didn't really anticipate how complicated it would be. But I think they don't make it easy because first you have to get the money right, and technically the SNF, the funding agency, wants to only give money to projects that are going to be ethically approved. So in the best possible circumstance, you would have the ethics approval first and then ask for the money. And that was of course impossible for me because my timeline is too short. So it's a lengthy procedure, and I believe it's appropriate that it's scrutinized at every step, but there have been very, very few cases in the world where things went amiss, when volunteers were harmed. So I feel confident that it's pretty safe. Yes.
Maybe I missed it, but how long does that procedure take until you are ready to actually start a project?
Well, in my case, it took it took almost a year to get the ethics approval. And probably if I'd been more organized about it or something, it could have been done in 9 months. But it just takes time because they almost always send it back for anything involving human research is going to be scrutinized in that respect. And then having received the ethics approval, then there's the separate entity, the BAG, which is a national organization, not a cantonal, and they have to approve it as considering the medical aspect of it rather than the pharmaceutical. It's the medical, the approval for administering a substance that is not in the pharmacopoeia, because this mixture of DMT and harmine that's being formulated in Zurich is not yet part of the pharmacopoeia, so it's giving a really experimental substance to people. So there's a high level of caution, but we're hopeful that it will be approved promptly because they've already received approval for a similar study in Zurich using the same formulation.
I do have another
Yes.
right at the start, you had this the slide where you said about the measurement of subjective effects, and you said you can only get so high. But I would tend to think that also that might have to do with the instruments. I mean, if you use the mystical experience questionnaire or whatever, I mean, it's very difficult.
Well, once you're one with the universe, you can't be more than that.
Well, I mean,
Sorry. Sorry. No. Yes. I mean, they use it's called the Likert scale, right? 1 to 10, and there's no real reason. It's very arbitrary. They use it for things like pain and on a scale of 1 to 10, and we don't know that it's linear. That the gap between 8 and 9 is the same as the gap between 9 and 10, and it's certainly reaching a kind of a plateau effect. Yeah. So the instruments are hard to pin down, but it will always be so. I mean, the physiological indices could be, obtained with some certainty, and the serotonin markers and things like that. But reporting on an internal state is always going to be problematic, I think. Even for psychologists. Any other questions? Yes.
We saw that psilocin triggers the release of dopamine without even acting on the dopamine receptor itself.
That's an interpretation, but no one else has investigated this, so we don't really know what's going on.
Could it just be that the trigger of the dopamine release is the mystical experience itself?
It depends if you're a dualist or not. Or if you believe in top-down effects. Yes, it could be. I don't know how to answer that question because I'm kind of schooled in pharmacology, but yes, there could be circuitous routes where your sort of mental state is feeding back on itself in some way, but not in a simple pharmacological sense. Yeah, it's a good point. I don't know how to get at that. Okay.